Chapter 13, Topic 3.1, Specific Immunity, the third and final line of defense. For this topic, I want you to be able to describe how the third line of defense is different from the other two, list the 4 stages of a specific immune response, discuss the role of cell markers in the immune response, and describe the major histocompatibility complex in 2 sentences. We're going to be covering lymphocyte development, presentation of antigens and clonal selection, the role of markers, specifically MHC and lymphocyte receptors, challenging B and T cells, the release of antibodies, and cell-mediated immunity. So in this topic, we're going to be doing a very broad overview of the adaptive immune system, and we're going to start with lymphocyte development. All lymphocytes arise from the same basic stem cell type. At some points, genes are turned on or turned off, and the cell goes down the path of becoming either a B cell or a T cell. The maturation process occurs in the bone marrow for B cells or the thymus for T cells. That's why they're called B and T cells. After they mature, but before they meet up with their specific foreign protein or antigen, they both circulate through the circulatory system, lymphatics, and lymphoid tissues. When pathogens breach the first and second lines of defense, phagocytes phagocytize the pathogen. The phagocytes then go around showing their antigens to T cells. Some soluble antigen can reach the B cells, but most B cells contact their antigen through contact with the T cell. Let's pause here to discuss foreign versus self antigens. All organisms need a way to tell self from non-self, or who is a member of the club and who is sneaking into the boys-only clubhouse. Mammals have a class of molecules called major histocompatibility complexes, or MHC. They are like cellular ID cards. All nucleated cells in your body have MHC1. MHC1 shows that the cell belongs in the body. Also, the cell displays some of whatever it is making on the MHC1. This way, if it is infected with a virus, it can display viral proteins, alerting the immune system that all is not right inside. MHC2 is found on immune cells such as macrophages, dendritic cells, and B cells. MHC2 is kind of like a police badge, telling other cells that it is part of the immune system and it's okay to be frisked by it. There's MHC3, but we won't worry about that one in this class. The MHC molecules vary from individual to individual based on a bunch of genes you get from your parents and are how the body recognizes itself. So back to B cells and T cells. The T cell shakes MHCs with the antigen presenting cells, which activates the T cell. Then they go and shake hands with the B cells. This activates the B cells. Activated T cells and B cells divide, making exact copies of themselves or cloning themselves. Some of these clones become memory cells so that the body can remember that particular pathogen. The non-memory cells go into battle. The B cells become plasma cells and make antibodies. The antibodies enter the blood. We call this humoral immunity. Activated non-memory T cells become one of 3 types of T cells: helper, regulatory, or cytotoxic. This is cell-mediated immunity. So that is our brief overview of the adaptive immune system. We will be discussing all of these steps in greater detail, so don't despair if it doesn't make any sense yet. Press on! Now, let's do some reminders of what I want you to be able to do now. Describe how the third line of defense is different from the other two. List the 4 stages of a specific immune response. Discuss the role of cell markers in the immune complex. Describe the major histocompatibility complex in 2 sentences.